Good morning, everybody. My name is Ronald de Groot. I'm president of the Dutch Foundation Save a Child's Heart. And I heartily welcome all of you. We're with a large group of medical students from both Europe and Africa, and one actually from another continent. And it's a delight that you're all here to be present at this first day of the virtual internship 2022. Before I introduce myself, I just want to spend 1 minute on the current situation in Ukraine. Being a pediatrician, and I know all of you being medical students, I'm sure that our hearts at this moment go to the children and the adults, both in Ukraine, but also those who are fighting a useless fight on behalf of the Russian Federation. So let's hope that this will stop as soon as possible and that wisdom will prevail. Having said that, the reason that I'm president of the foundation in the Netherlands is that I've got a long career in pediatrics with a strong interest in global health. So how can we help children in this privileged situation? And this is exactly how SETS was started 25 years ago by a pediatrician in Israel, Amy Cohen, who saw that children in Ethiopia were not able to get appropriate care for those who had a congenital heart defect. And as all of you know now, approximately 1% of all children in the world have congenital heart defects. And unfortunately, many of those children can't get appropriate care. They can't get appropriate care for pediatric cardiology. They can't get surgical interventions. They can't get other types of interventions and adequate therapy, so that they die very early. And this is exactly the mission of Save a Child's Heart, which was founded in Israel. We have now 5 or 6 foundations all over the world: the United Kingdom, USA, Australia, Canada, Switzerland, and our foundation in the Netherlands, which has taken the initiative to do this virtual internship, where we will have 6 courses in the coming months. We've had a large number of participants, 160 approximately, and for this first course, we're very grateful that you all were willing to present. So, having said that, we also have a very active young group of physicians and other people who are involved in Save a Child's Heart. And my co-chair today, Emma Oosterbroek, will take over later on and tell you a bit about the activities of young Save a Child's Heart in the Netherlands. Obviously, we try to get money to do the good work in Israel, where children are brought from Africa and from the Middle East and from other continents to be operated, where we also have a facility where the children can stay with their parents until they're operated and after the operation. So this is a wonderful project, which actually has had a prize of the United Nations as one of the most humanitarian projects in the world. So having said that, Emma will start now a video which will give you a short introduction to our activities. And then Emma will take over, introduce herself, say a few words about the procedures, and then she will also introduce the first speaker. So I hope you have a very good meeting today. The 2 of us will be here all the time to answer questions. Obviously, the speakers will answer all your questions. And we're delighted that we're with so many people for such a fantastic goal to how can we help kids in these privileged situations who need cardiac care.
Around the world, 1 in every 100 children is born with a congenital heart defect. 25% of those children will need medical care to survive. Hundreds of thousands of children develop heart disease in their lifetimes.
My name is Kasema Maria Tendo. I'm an 11-year girl from Uganda. When I was 3 months old, my mother thought I had a heart problem. And when we went to the hospital, it was true. We were connected to Save a Child's Heart in Israel. I had first come here when I was 9 months, 6 years, and now I'm here again. Save a Child's Heart gives me hope.
For over 25 years, Save a Child's Heart has worked to save children, train medical professionals, and raise the level of pediatric cardiac care worldwide. To date, Save a Child's Heart has saved the lives of over 6,000 children from more than 60 countries around the world.
It's a great privilege for me to work here as a pediatric cardiologist for Save a Child's Heart. Half of the children we see are the Palestinian children, the other half of the African children. The Palestinians we see here in Israel in clinic, in hospital every Tuesday, regardless of the political situation in the area. On the other hand, the African children are far away. So at least 3 times a year, each of the pediatric cardiologists goes abroad on a mission to screen the children in Africa. I'm touched on a daily basis.
Over half of the children we treat are Palestinian children from the Palestinian Authority and Gaza. Since 1996, every Tuesday, Save a Child's Heart has hosted a free clinic to screen, diagnose, and follow up with Palestinian children who've received medical care in Israel.
After so many years of conflict, of course, people get frightened, they don't know the other side. So when they get here, they see how much Save a Child's Heart care about them and they get the help, so the fear disappears. And at the end of the day, they see that we all are people and human beings.
But our mission goes beyond treating children. Our goal is to improve the level of pediatric cardiac care all around the world. At Sylvan Adams Children's Hospital and the Save a Child's Heart International Pediatric Cardiac Center in Israel, Save a Child's Heart brings medical personnel from partner sites around the globe to train in all aspects of pediatric cardiac care. With heartfelt courage, we are building a better future for all children.
I am Stella Mongela. I'm a pediatrician from Tanzania, and I am currently in Israel training in pediatric cardiology. It is a 2-year training program, and this is my 2nd year. The need for pediatric cardiac care in Tanzania is great. We have a country of 60 million people. We have about 2 million new babies born every year, of whom 16,000 statistically would be born with heart disease. My hope for the future is for the advancement of pediatric care in general, and of course pediatric cardiac care. And I'm glad to be in a position to be able to contribute somehow to helping to ensure that Tanzanian children grow up to be very healthy and to be productive Tanzanian citizens.
Together, the children, their parents, our partners and trainees, the medical team in Israel, and our worldwide family make up the community it takes to save a life. And to save a life is to save the world. Help us bring hope to children and their families. Join our mission today.
Every time that we can say yes to a mother, not a conditional yes, but an unconditional yes, bring your child and we can provide him what the 21st century has to offer, I reflect on what we were 17 years ago, what we're doing today, and how we're doing the right things with the things we've obtained. We at Save a Child are proud to do our share in this metamorphosis and are deeply grateful for your support that allows us to continue and expand our work.
Hello, everyone. So my name is Emma. Let me introduce myself first to you. Together with Ronald, I live in the Netherlands and I'm part of Save a Child's Heart in the Netherlands. and I'm in Young Leadership, the Netherlands. So together with young leaders around the world, we work on raising awareness for Save a Child's Heart, raising funds, and together we are improving international cardiac care for children worldwide. I'm so excited to see you all here today. Together with so many international medical students, we are going to join in this internship with 6 interactive workshops. Today we have 2 speakers joining us, and we are beginning at the start of it all. So at first we will focus on embryonic cardiovascular development, and after our break we will focus on the anatomy of the heart. Let me ask you to please turn on your camera. During the presentations, please mute, but we would definitely like to encourage you to join the discussions during the Q&A, to connect with each other during the breakout rooms that we will open in between both sessions. Because the aim of this international internship is to connect and to improve international cardiac care for everyone. So please feel free, and we would definitely encourage you to connect with each other and ask your questions. To do so, please use the chat or raise your hand, and when there is time for questions, I'll point out who can answer your questions. Don't be shy, please ask all your questions, because that's what we're here for, and I would definitely encourage you to join the discussions. Let me see, because then I will continue for our first speaker. I would like to introduce Catherine Roberts. She's here with us today. There she is. At first, she had her degree in genetics and cell biology at the University of Manchester, and then had a PhD in Oxford on embryology and investigating molecular mechanisms of gastrulations. And that led her into the field of cardiovascular development biology, and is working on the cardiovascular aspects of the 22q11 deletion syndrome. At this moment, she's a lecturer in medical sciences at the Institute of Medical and Biomedical Education at St. George University in London, and will present for us the cardiovascular development, the heart fields today. Catherine, would you like to take over?
Sure. So hi, everybody. It's great to see so many people here, and I'm very excited to be taking part today. So today I'm going to talk to you about cardiac developmental biology, and I'm going to mostly focus on cardiac morphology to start with. If we have time, I'll get into the genetics a little bit, but we'll start off with the morphology. So just let me try to share my screen. Okay, so today I'm going to talk to you about congenital heart defects, cardiac development and regeneration. So as Tamer's already mentioned, around 1 in 100 children are born with a congenital heart defect, all sorts of types affecting many different structures of the heart. And today I'm really going to sort of start by quickly recapping different types of congenital heart defects, but I'm sure this audience probably knows perhaps more about the sort of surgical and medical side than I do. So then I'm going to concentrate on the cardiovascular development that underlies the normal development of the heart, that allows it to move from, so these are mouse embryos, picture of a mouse embryo, and a lot of my pictures will be mouse embryos. So, because that's a common model in the laboratory. And I'm going to be talking about how we go from a small ball of cells with 2 cell layers to, within 7 days in the mouse, to a 4-chambered beating, pretty much mature heart. And how, when that process goes wrong, it gives rise to the congenital heart defects that we see in both our animal models and in human patients. If there is time, I'm going to talk a little bit about how developmental biology informs cardiac regeneration and repair, and perhaps share a few slides on the sort of research projects that I've undertaken and I'm still working on over the years. Not quite sure how the timing of this is going to work, so I'm, may sort of mix and match a little bit, perhaps, depending on how time goes, see how we're doing. There'll be little sort of pauses along the way, so you can ask questions there, or you can leave your questions to the end. It's fine, it's up to you. I'm not sure that I can see the chat while I'm sharing my screen. So, if you put questions in the chat, I'm not sure I'm going to be able to see them until I've finished screen sharing. Okay, so talking a little bit about congenital heart defects. So obviously you're going to recognize this. This is a schematic of the normal heart with deoxygenated blood coming in, being pumped out to the lungs to be reoxygenated, come back into the left ventricle and being pumped out through the aorta to the rest of the body. So when we think about congenital heart defects, we need to define them. And really a very basic definition is that there are defects in the structure or the function of the heart and the great vessels, which are present at birth as a result of abnormal embryonic development. And this can be caused by a number of different factors, generally genetic, but sometimes environmental, and sometimes we actually are unable to uncover quite what's caused the problem. So congenital heart defects account for about 10% of spontaneous abortions, and they affect nearly 1% of all newborns, and they are the most common birth defects. The severity of a congenital heart defect is very variable. Some are very simple, some are very complex, and the mortality rate is therefore also very variable. So in previous years, being born with a congenital heart defect, particularly some of the more complex ones, would almost inevitably have led to death early in childhood. However, we now have a range of remarkable surgical and medical advances that are allowing nearly 90% of surviving children to live into adulthood. And this is establishing the field of adult congenital heart defects as people live longer with these paired defects. And we're learning more and more about how we need to support these patients to continue living their lives into adulthood. So major underlying defects causing these defects are mutations in genes that regulate heart development during embryogenesis. And one gene mutation can produce multiple types of congenital heart disease because one gene can regulate the development of many different elements of the heart. Alternatively, you can get one type of congenital heart defect being caused by mutants in very many different genes, and this is because of the pathways of genes that are involved in regulating parts of cardiac development. They can present in isolation or as parts of syndromes, which can affect other developing organs. And in addition to genetic causes, epidemiological studies have shown that maternal infection, so for example, rubella, drugs, alcohol, maternal illnesses, for example, diabetes, maternal obesity, and so forth, can also have impact on the development of the heart of the fetus. So this is just giving you a little overview of where we find congenital defects, and it shows incidence per 1,000 live births. And you can see that we find defects sort of throughout the structures of the heart. There are hotspots. So the great vessels, sorry, my pointer disappeared. The great vessels are particularly prone to developmental defects, as are the septum and valves of the heart. But these are not the only places where we find defects, and we'll go on to discuss some of this in more detail. So we can classify congenital heart defects into three broad categories: cyanotic heart defects, left-sided obstruction defects, and septation and valve defects. And I'm just going to run through a few quick examples of these, but try not to spend too much time on this because really we want to talk about the development. So cyanotic heart defects occur if the body tissues are not receiving properly oxygenated blood. So if you're getting mixing of oxygenated and deoxygenated blood, and these defects affect cardiac great vessels and septation. So things like common arterial trunk, tetralogy of Fallot, double outlet ventricle, and so forth. So these are just some schematics showing you a couple of examples. So here's a normal heart. This is common arterial trunk. The septation of the outflow tract has not occurred normally. And so the aorta and the pulmonary trunk are not separate, and you've got mixing of oxygenated and deoxygenated blood. And if this remains untreated, about 75% of patients will have died by the age of about a year. However, following surgical repair, we're finding that there's 95% survival, which is really impressive. Tetralogy of Fallot is quite a complex congenital heart disease, and it's quite rare. It's often associated with 22q11 deletion syndrome. And here, sorry, you get an obstruction affecting flow of the blood out the lungs, and there's usually also a ventricular septal defect and a hypertrophic thickened wall of the right ventricle. And again, untreated, the mortality rate is 50%, but again, there's a very high success rate following surgical repair. We also see things like double outlet right ventricle, where rotation of the heart during development does not occur normally. And this means that the great vessels don't shift to exit the ventricles as they should. So you end up with both the aorta and the pulmonary trunk coming out of the right ventricle. So you don't get pumping of oxygenated blood around the body. And there is a variety of subtypes depending upon where the associated ventricular septal defect is found. So left-sided heart defects affect the function of the left side of the heart in pumping oxygenated blood out through the aorta. And these include left ventricle and aortic valve defects, so things like hypoplastic left heart syndrome, interrupted aortic arch. So hypoplastic left heart syndrome is a very, very severe defect. And basically the left side of the heart is extremely underdeveloped with a tiny left ventricle, which means that there's a real lack of oxygenation. And it's often fatal without treatment, often in the first day of life. And it's a very complex surgical repair, and it's really palliative only. And this remains one of the syndromes we're still, despite quite a lot of research, still not really sure what the underlying causes are. So interrupted aortic arch is relatively common, and some of types of it are particularly associated with syndromes such as 22q11 deletion syndrome. So this is the normal aortic arch with the great vessels coming off it, joining the ascending and descending aorta. However, we get these defects where we get a break at some point along the aortic arch. Now, during embryogenesis, the embryo can survive because circulation can be sent via the pulmonary duct. But once the pulmonary duct closes after birth, then an interruption is lethal. And they get subdivided into different types depending on where the point at which the break occurs. So type A occurs here, just below the left subclavian. Type B, which is the commonest type, occurs between the left common carotid and the left subclavian. And it's these cases always have an associated ventricular septal defect. And this is strongly linked to 22q11 deletion syndrome. And type C, which takes place just beside the right common carotid, is very, very rare. And surgery is typically performed within the first few days of life to reconnect the aortic arch and repair the VSD. So there's also a sort of spectrum of lesser similar types of defects called coarctation and tubular hyperplasia, which are narrowing of the aortic arch at various different points, and quite how the names depend on the position of the narrowing or the stenosis of the aortic arch. And then finally, we have septation defects, which affect the division of the heart into the four chambers, and they frequently will lead to mixing of oxygenated and deoxygenated blood, but they're less severe than in cyanotic defects. So we have atrial septation defects, ventricular, and then both. and also defects affecting the valves where the number of valves leaflets is altered, or they're too small or too large and don't function normally. And these can be seen in isolation or combined with other defects. And in isolation, they're fairly minor and can be easily repaired surgically. So just a quick schematic to show you, I'm sure this is the sort of thing that we're quite familiar with. So, atrial septal defects, we get a break in the atrial septum, ventricular septal defects, a break in the ventricular septum, and the position at which the break occurs affects how we categorize them. So breaks up here, close to where the ventricular septum is fusing with the atrial septum, so forth, will be called perimembranous, and these further down in the more muscular septum are called muscular septum. Sorry, down here. And then in an AVSD, we get defects affecting both the atrial and the ventricular septum, and frequently affecting the mitral and tricuspid valves. And again, this leads to, as you can see here, sort of common valve with no divisions, mixing of oxygenated and deoxygenated blood, increasing problems such as congestive heart failure and so forth. Heart valve problems tend to fall into two major categories. So stenosis, where the valve is too narrow, and regurgitation, where the valve doesn't close properly, leading to backflow of blood. And the valve leaflets, as I said, can be altered, altered in number, or too big, too small, which means that they don't close properly. Sometimes, often genetic components in this, and valve defects tend to be one of the most common types of congenital heart defect that we see. And it's also important to remember that defective valves can also increase the likelihood of calcification and atherosclerosis of the valve later in life. So, this is an ongoing effect through the patient's life. However, it is relatively easy to repair or replace valves. So this is quite a common surgical procedure. And then very quickly, we also have congenital defects that affect the way the heart beats. So conduction of the electrical impulses, which regulate the heartbeat, lead to arrhythmias. And the disconnection between atrial and ventricular conduction, and therefore contraction, can lead to abnormally fast or slow heartbeats, leading to abnormal blood flow. And then just to mention, so that you're aware of them, although I'm not going to focus particularly on this, there are defects which will be present from birth that affect the contractile muscle of the heart, and these are cardiomyopathies, and there are several different types. And we can also have defects in cardiac vascularization, where the patterning or formation of the coronary vessels are affected. And if I have time, I'll also talk a little bit about the link between developmental biology and recovery from a heart attack. So just to sort of reiterate, we get cardiac defects at lots of different parts of the heart. But why is this? I'm just going to take you through cardiac development. First of all, thinking about cellular morphogenesis, and then thinking about some of the genetic networks and the disease that can arise. So obviously this is a very, very complex area. So this is really just going to give you a sort of very brief overview. Okay, so cardiac development actually starts really early. It actually starts at gastrulation, which is when you make the mesoderm of an embryo, which arises from the ectoderm following signals from the endoderm. And this is at E6 in the mouse, so about a third of the way through gestation, day 13 to 14 in the human. And we get the exit of mesoderm. It's sort of midway through gastrulation, and it's coming out of the mid position of what we call the primitive streak, which is the first sort of axial structure of the embryo that will eventually give rise to the notochord. Sorry, I'm having a bit of difficulty with my mouse. Okay, there we go. And on either side of the midline, we're going to have we're going to become cardiogenic mesoderm exiting the primitive streak and comes to lie more anteriorly within the embryo, which you can see here about a day later, forming these two crescents, which are two different types of cardiogenic mesoderm. One in red is called the first heart field, and the one underneath it is called the second heart field. And these are going to give rise to most of the cardiomyocytes of the heart. We're also going to get the addition of additional extra non-cardiac cell populations, which are nonetheless super important for the development of the heart. By 14.5 days, there's going to have been a huge amount of remodeling, so that we've gone from this to basically a 4-chambered beating heart with totally remodeled great vessels that in most respects is extremely similar to the adult heart. There are some remodeling that takes place after birth, but most of the pattern of the heart is already laid down by this point, which is 2/3 of the way through mouse gestation and at day 32 in the human. So then, you know, obviously this is a big change in a really short space of time. And there are a large number of genetic and molecular and cellular processes involved. So obviously genetic activation and repression, regulation of transcriptional networks, epigenetic control of chromatin status, signaling pathways, proliferation, apoptosis, balancing progenitors and differentiation, epithelial mesenchymal transition, cell migration. You can go on and on and on. And we're going to talk a little bit about some of these processes and how they're important in cardiac development. So understanding this allows the discovery of mechanisms which lead to congenital heart defects and can provide possible therapeutic directions, although that's something that's still very much under investigation. But also understanding how these defects come about, modeling them and so forth can also help surgeons come up with more efficient surgical repair. And it's important to understand that cardiac anatomy, morphology, genetics, and cell lineage, fate, migration, signaling, they are all integrated in development. So when we're investigating cardiac development, we use a number of different strategies. These include a lot of animal models, because obviously working with human tissue is not always possible. The mouse, the chick, the zebrafish invertebrates have all been used. We work with human patients. We also work with termination material. And increasingly, people are using embryonic stem cells and infused pluripotent stem cells as models. So a heart in a dish, if you like. And all of these have advantages and disadvantages as models. And it's important to note that we can use these animal models because of evolutionary conservation. So the programs that regulate the development of the heart in other vertebrates or even in lower species and invertebrates are often the same as in humans, and we can learn information that's relevant to the human from these models. So talking a little bit about cardiogenesis. As I've said, the mesodermal layer induced from the epithelium by signals from underlying endoderm during gastrulation will give rise to the cardiac mesoderm, and this will give rise to all the myocardium of the heart. So this is just another little model showing you the cardiac mesoderm coming out of the primitive streak in a mouse embryo. And a mouse embryo is a bit of a cylinder. The primitive streak forms from the anterior to the posterior region here. And in green, you can see the cardiac mesoderm moving out, moving anteriorly, and eventually forming the cardiac crescent. So this cardiac mesoderm will give rise to atrial and ventricular cardiomyocytes, also endocardial cells, which will eventually go on to become more specialized cardiac cells, including cushion cells and conduction cells. if we look at the top panel, you'll find that the first heart field gives rise to sort of what we call the linear heart tube. So that's the first part of the heart to form, and it mostly goes on to give rise to the left ventricle and parts of the atria. And the rest of the heart is formed by the addition of extra cell populations. And the first of these is the second heart field, which is added after the formation of the linear heart tube. And it gives rise, it's added on at the arterial and the ventral poles. And this gives rise to the outflow tract and the right ventricle, and also contributes to part of the atria. Then a bit later in development, we need the addition of two non-cardiac cell populations to allow the heart to develop as it should. And the first of these is the cardiac neural crest. So neural crest migrate from the dorsal neural tube along the entire axis of the forming embryo. The cardiac neural crest come from the region of the posterior hindbrain and anterior somites, and they migrate through these structures called pharyngeal arches, which I'll talk a bit more about later, and they move into the septum of the heart, where they're required to form the muscularizing septum, which divides the outflow tract and the aorta, and divides the outflow tract into the aorta and the pulmonary trunk. And they're also necessary to provide the vascular smooth muscle that supports the endothelium of the pharyngeal arch arteries, which are going to remodel to give rise to the great vessels. The second population is the proepicardium, and this is this little ball of cells that forms at the base of the heart, just below the atrioventricular groove, but it's not actually part of the heart. This happens quite early in development, around E9.5. We find that the cells here migrate out of the proepicardium, stick to the myocardium, proliferate and spread out and cover the entire surface of the heart. And they then undergo epithelial mesenchymal transition, move into the myocardium, and they're required to contribute to the lineages of coronary vasculature, so smooth muscles of the coronary arteries, also the endothelium of the coronary arteries. And it's also, epicardial cells are also extremely important for giving rise to cardiac fibroblasts. So thinking about our two heart-forming regions, the first heart field and the second heart field, as I've just said, they contribute to largely different regions. And we think of the first heart field as the scaffold of the heart, and the second heart field as being added afterwards to the arterial or anterior or venous / posterior poles of the heart. And this slide basically just recapitulates what I've just said. So let's think about the first heart field a little bit more. So here you can see this in red, and this is showing you the structures that the first heart field will give rise to. Unfortunately, the little video panel is blocking your view of the final part here, but it's giving rise to the left ventricle and parts of the atrium. And here we can actually see the first heart field in mouse embryos as it forms, because we're using a myosin light chain reporter, where basically myosin light chain is specifically expressed in first heart field cells, and we're using it to drive the expression of a LAXI reporter. And then when we develop that reporter, we can see where first heart field is at different stages in the embryo and which cells it's contributing to. So here we can see in the cardiac crescent, and then in the linear heart tube as it begins to develop. So moving on, that's quite brief on the first heart field, and I'm going to focus more on the second heart field, partly because that's where a large number of congenital heart defects arise, and we actually understand more about how congenital heart defects linked to the second heart field come about. But I will talk a bit more about the first heart field later on when I talk about molecular control of these different pathways. So the second heart field, which is shown here in green, is a reservoir of progenitor cells in pharyngeal mesoderm, and it's added progressively to the first heart field. And the arterial addition at the outflow tract is shown in dark green and the venous addition in light green. And these cells are characterized by continued proliferation and a differentiation delay, which allows them to add in sufficient numbers to the poles of the heart to allow the normal development of the heart. And if something goes wrong with that, then the remodeling of the heart will be affected and will end up with congenital heart defects. So again, this is a reporter. This is a lax enhancer trap of FGF10, which is important in regulating second heart field development. And you can see the second heart field crescent, the additional adding of cells to the crescent, then the second heart field cells start to migrate out, joining onto the linear heart tube. And you can see here in this side view, them migrating through the forming pharyngeal arches and eventually contributing heavily to the outflow tract and the right ventricle. So, you know, this continued proliferation and delayed differentiation allows the elongation of the outflow tract, which is important for normal rotation of the heart. And if that doesn't happen, you end up with defects such as double outlet right ventricle. And it's controlled by two different but related networks. First of this is a complex signaling environment in the pharyngeal region, where you've got signaling between neural crest cells and second heart field cells, and the signaling from a number of different known signaling pathways, say Wnt and beta-catenin, Notch pathway, fibroblast growth factors, BMPs, hedgehog, and they're regulating the balance between proliferation and differentiation. We've also got a distinct transcriptional program of transcription factors in the pharyngeal mesoderm, controlling the differentiation of secondary heart field mesoderm. And so these inductive signals will give rise to the expression of genes at the top of these pathways, so things like TBX1, islet1, which will in turn induce this sort of second heart field characteristic transcriptional network. And you can't see it under here, but the first heart field has got its own characteristic set of transcription factor signal expressions as well, regulated by genes such as GATA4 and GATA6. So, you know, this second heart field derived outflow tract rotation and alignment is reliant on the elongation of the heart tube by adding these proliferating myocardial progenitor cells. And as this is added, we also at the same time begin to have rightward looping of the heart, which allows the inflow and outflow poles to come together. So you can see early in development, outflows up here, inflows down there, and then as the heart loops rightwards and rotates, the outflow and inflow start to move much more closely together. And the outflow tract needs to be of sufficient length to allow this looping to take place robustly and to allow the myocardial wall of the outflow tract to rotate correctly. Because the myocardial wall needs to rotate counterclockwise before and during the formation of the great arteries. Because if it doesn't, then the great arteries don't go into the correct ventricles. And so this looping occurs concurrently with chamber development, so that atria and outflow vessels are then correctly aligned with the ventricles. And in genetic mouse models which have outflow tract alignment defects, this rotation is often abnormal, and it's often due to the underdevelopment of the myocardium at the base of the pulmonary trunk and also the length of the outflow tract. And TBX1, for example, is a gene that's important in regulating these processes, and we'll talk a little bit more about that later. So let's move on to sort of discussing cardiac chamber formation briefly. So, okay, we've made this linear heart tube, well, that's great. but you know, it's not a four-chambered heart. So how are we going to do that? So as the heart loops from left to right, and sort of ventrally, you get chamber myocardium expanding from the outer curvatures of the primary heart tube. So you can see this beginning to happen here, and then here. Again, this is mouse. And this myocardium acquires working myocardium characteristics, so it's capable of fast conduction. But through the center of the heart, this region here in gray, this remains non-chamber myocardium. So we have the inflow tract, sinus halls, atrioventricular canal, and so forth. And this remains as the more primitive type of myocardium with slow conduction. Excuse me. And some of this myocardium will give rise to conduction structures such as the atrioventricular node. It's also where the atrioventricular junctions will fall. So there are specific transcriptional regulators, so transcription factors such as ANF and other genes, which are expressed in chamber myocardium and not in primary myocardium. And they're required to allow this ballooning to take place. And you can see here expression of ANF in the forming ventricle, and then more strongly in the as the ventricular chamber develops and balloons out. Oops. And just to mention the sort of the T-box transcription factors are particularly important in patterning the heart, in forming building blocks of the heart here. And you can see in this schematic that TBX1 is important for the formation of the outflow tract. You need TBX2 and TBX3 in the non-working myocardium to form the conduction system. TBX18 in the inflow region and for epicardial and coronary development. And then TBX5 is very important for allowing normal chamber development. So a little bit more detail on pharyngeal arches, arch arteries, and cardiac neural crest, because these are intimately connected. So the pharyngeal arches are this series of transient embryonic structures, which in the mouse start forming around E8 and are pretty much gone by E11, 12. And they're these, anterior to posterior bulges that form in what's going to be the head, neck, and cardiac region. And they consist of different layers of tissue. So at the center of each one is an arch artery that's surrounded and supported by core mesoderm, which in turn is surrounded by cardiac neural crest derived mesenchyme. And these structures form around the pharynx, which is lined with endoderm, and on the outside they're lined by pharyngeal ectoderm. Now these bulges very much form because of the influx of neural crest from the dorsal neural tube, and they form in a anterior to posterior direction with time, and this is reflected by the migration period of the neural crest. So cardiac development, we have neural crests coming in, populating these bilaterally paired aortic arch arteries, giving rise to vascular smooth muscle which supports them, and then also moving into the outflow tract of the heart for septation. And you can see, so the different aortic arch arteries have been colored here. And you can see that from earlier development, the aortic arch arteries remodel greatly with quite a lot of regression of different parts of the aortic arch arteries to give rise to the mature great vessels. And you can see different parts derived from the different pharyngeal arteries colored here. And if we disrupt cardiac neural crest development, then we will disrupt this process. I hope this video is going to play properly, just so that you can see how complex this remodeling process is. There we go. So if we start again, so arch arteries 3 are going to give rise to parts of the carotids. Arch arteries 4 are going to be important in the right subclavian and the aortic arch. And arch artery 6 is important in the duct and the pulmonary arteries. And for some reason, and we still don't fully understand why, fourth pharyngeal arch arteries are particularly prone to defects. So abnormal development of the fourth pharyngeal arch artery on the right will give rise to an aberrant right subclavian defect. So it's often retroesophageal, but sometimes it's just missing. And on the left, we'll end up with an interrupted aortic arch type B, because the left fourth pharyngeal artery contributes to this region of the aortic arch. So, as I've said, neural crests are particularly important for this remodeling and for the septation of the outflow tract. And you can just see here in this picture, this is PAX3, which is a neural crest marker driving a fluorescent reporter. You can see the neural crest migrating out, and then you can see them coming into the outflow tract of the heart here. And they populate the outflow tract cushions there, giving rise to the septum, and as I've said, are important for normal rotation. So the outflow tract cushions form in a spiral arrangement to each other, and they expand and fuse together via endothelial mesenchymal transition as neural crests migrate into the cushions. And this is then followed by second heart field smooth muscle, leading to the formation of the myocardial septum. And ultimately, this allows the normal septation of the outflow tract into the aorta as they spiral around each other. And you can see here the neural crest shown in green being intimately involved at the center of the septating outflow tract, being required for the formation of this muscularized septum. And eventually, this forming myocardial bridge will resolve, and you'll end up with the aorta and the pulmonary trunk completely separate. So none of this really happens in isolation. And this slide is really just to show you that there's a huge amount of crosstalk and interaction between the second heart field and neural crest cells that are important for outflow tract development and pharyngeal artery and great vessel development. And that if this is disrupted at a number of different points, then you could potentially end up with congenital defects. And just a quick look at the pericardium and epicardium. So these are scanning EMs, and you can see the pericardium, this little ball of cells at the base here, spreading out and covering the heart. And eventually, some of these cells, under the influence of secreted signals such as Wnt's, FGFs, retinoic acid, will undergo EMT, give rise to cells that migrate into the myocardium and produce cardiac fibroblasts, parts of the coronary vessels. And there's some sort of controversial evidence that they might also give rise to cardiomyocytes. The epicardium is also important because it sends secreted signals to allow myocardial proliferation and compaction. So if we move on to thinking about the genetics involved here, and this is not going to be in any way comprehensive because we'd be here forever, but just to give you a little flavour of what's going on here. So this schematic basically shows you the progression from a pluripotent stem cell through to differentiated types of cardiac cells. And we move from a pluripotent stem cell, which will express factors associated with pluripotency, such as OCT4, SOX2, KLF4, and so forth. And what we'll get is signaling from a number of different secreted signaling pathways, so BMPs, activins, and Wnt's, which will induce the expression of genes that are associated with being a primordial cardiovascular precursor, in particular MESP1 and FLIC1. So this is done to move these cells down a differentiation pathway towards being cardiac mesoderm. We'll then get further cardiac specification by very similar but reiterated signals from these pathways. So again, the MPs, activins, FGFs, non-canonical Wnt's, and we actually need inhibition of some canonical Wnt signaling here. This will lead to the production of first and second heart field cardiac progenitors, which will express specific gene programs. So first heart fields will be expressing transcription factors like GATA4, AKX2.5, whereas the second heart field will express ILAT1, DBX1, MEF2C, and so forth. And there's a set of secreted signals which are important for maintaining these progenitors and allowing the heart to continue to grow. As we then move through development, we'll get another set of secreted factors, again, very similar to, oops, sorry about that, very similar to the factors that we're signaling earlier, but this is taking place later in development, so it will have a different effect, which will allow the differentiation of different specialized types of cardiac cells. So ventricular cardiomyocytes, atrial cardiomyocytes, pacemaker cells, and so forth. So just to show you some of the studies that have led to some of the understanding that we have of these processes, let's think about MESP1, which is one of the main regulators of precursor cells. So if you knock out MESP1 on its own, you will affect cardiac development quite badly. And actually what you end up with is cardia bifida, so the formation of two hearts. And this is because MESP1 is important in regulating the migration of cardiac progenitors of the heart fields. However, there's a second MESP gene, MESP2, and this can compensate for the loss of MESP1. And this is something that we see quite frequently in genetic studies, that we will get compensation or genetic redundancy when we knock out one gene, and sometimes we have to knock out a second gene to really understand what's going on. And this is what we find here. When we knock out MESP1 and MESP2, we find that cardiac mesoderm doesn't form. So you can see here normally formation of mesoderm, but this just does not happen in the mutants. And we also see in humans that there are genetic variants and mutations associated with MESP1, not normally null mutations, because probably those embryos just won't survive. There'll be spontaneous abortions. And mutations in MESP1 in humans tend to be associated with severe congenital heart disease, such as hypoplastic left heart syndrome, but also defects arising from abnormal development of the second heart field. So don't worry about the detail on this. This is just to show you what an important regulatory role MESP1 has. So, you know, it's pan-mesodermal and it's required for migration of cardiac progenitors. And you can see at the top of the schematic, it's regulating a whole, in a positive fashion, a whole host of genes associated with onward cardiac development, from TBX5 for chamber development, F2C necessary for second heart field, GATA4 necessary for cardiac progenitors, and so forth. In addition to this, it's also got a negative regulatory activity where it's going to repress genes associated with endoderm and blood formation. So it's playing a multiplicity of different roles during cardiac development. This is sort of studies from both mouse and chick that is showing you the signaling pathways important in actually inducing heart formation. So you can see here we've got expression of BMPs around the outside of the embryo, Wnt inhibitors at the top, Wnt signaling at the bottom. And these signals come together to regulate the formation of the heart versus the formation of blood. So if you send a BMP signal towards mesoderm, you're going to be potentially inducing both heart and blood. If this tissue also sees a Wnt signal, it will definitely make blood. However, if you inhibit the Wnt signal and the tissue only sees the BMP signal, then you'll get the formation of a heart. So you can see that it's important to regulate which tissue sees what signals at what particular time. These signals will induce the expression of cardiogenic transcription factors such as GATA4, NKX2.5, and MEF2C in heart-forming regions. And this just sort of shows you some of the experiments that were done in Xenopus to try and prove this, where if you overexpress Wnt signals, then you'll block cardiac formation. Whereas if you inject Wnt antagonists ectopically, then you can actually induce the formation of beating heart in a place that you wouldn't normally. You can see here. And then also to remind you that later in development, Wnt signaling regulates the expansion of second heart field progenitors through islet-1 and FGF signaling. So you can see here, this is normal. This is where Wnt signaling has been experimentally increased, and you've got this big expansion of second heart field progenitors. And then this slide is just really to sort of show you how important BMPs are in cardiac formation, because basically you knock out a lot of cardiac ligands, cardiac receptors, sorry, not cardiac ligands, BMP ligands, BMP receptors, and their downstream intracellular effectors. You'll end up very often with no mesoderm formation or a small heart because of reduced proliferation or other cardiac defects. Okay, so just to remind you about heart fields, second heart field, first heart fields, and revisiting our regulatory schematic. And then to sort of say that schematic, this same genetic network that you can see here that's regulating these heart fields is also present in invertebrates such as Drosophila. This is a highly evolutionary conserved network. And these genes up here are linked to the first heart field in mammals, and down here then to the second heart field in vertebrates. And you can just see up here the Drosophila heart with an aorta and the heart, and it's just a single tube. But this core regulatory network still controls the development of the heart, and those genes still important in much more complex organisms. Excuse me. So one of the genes important in cardiac development in generally, but particularly for the first heart field, is GATA4. And GATA4 mutations in humans are associated with a variety of different defects. And when we knock it out in the mouse, again, we see severe cardiac defects such as cardia bifida, reduced myocardium, double outlet ventricle. Now, these defects implicate GATA4 in cardiomyocyte proliferation and differentiation, and it has a general role in progenitor cell expansion, in addition to roles in endothelial cell function and in epithelial mesenchymal transformation necessary for forming cardiac cushions for valves. NKX2.5 also is being shown in animal models to be very important for normal cardiac defects, sorry, normal cardiac development. And you can see here that, you know, in a normal mouse at around 8.5, you've got a nice formation of a linear heart tube. It's starting to loop, whereas in the mutant, it's just sitting there. It's not looping. It's not showing any signs of you know, really ballooning. and you really don't have the beginnings of the formation of either ventricles or the atria. And in humans, NKX 2.5 mutations are associated with a variety of structural defects and valve defects, and also with conduction defects, because this gene also regulates the conduction system. And then looking at TBX5, which we mentioned earlier as being really important for cardiac ballooning, you can see here's a normal heart labeled up, and in the mutants, it's very, very much reduced in size. And you can see very much reduced formation of ventricular development and ballooning compared to normal. Oops. Again here, very, very small heart formation compared to normal. Mutations in TBX are associated with Holt-Oram syndrome, which affects the heart and also the upper limb. You get septation defects, trilogy of Fallot. It's also associated with hypoplastic left heart. And if you knock out TBX5 in mice, then you're basically embryonic lethal, because the atria fail to develop properly, ventricular development is also affected, and you get a great reduction of specific chamber genes. So you don't form normal cardiac chambers. So this slide is just to show you, so that's first heart field. And we're now moving on to think about second heart field and cardiac neural crest. Now, that is a very complex network, genetic network. So this is really just to show you some of the genes that we know are involved in second heart field development and that we know potentially lead to human defects. So for example, TBX1 is implicated in 22q11 deletion syndrome, CHD7 in charge syndrome, Notch in Alagille syndrome, and so on and so forth. And really the take home from this is just how complex this process is and the huge amount of crosstalk that's going on between the different tissues, how they all have to express the right genes at the right time, signal to each other at in the right place and the right time to allow cardiac morphogenesis and development to proceed normally. And this simplifies that a little bit, allegedly. And just, the top panel is essentially about TBX1 and islet1 regulating the second heart field proliferative progenitor state. They then also regulate, along with beta-catenin Wnt signaling, FGF and BMP expression. And there's an antagonism between FGF, which is pro-proliferation, and BMP, which is pro-differentiation, which is really important in controlling the balance between proliferation and differentiation in the second heart field. Because if this gets out of sync, then again, we'll end up with cardiac defects. And eventually downstream of all of this, we get differentiation and activation of cardiogenic program and a network of different interacting transcription factors regulating the myocardium. And I think having seen this, you can understand why at least 30% of congenital heart defects arise from perturbations of the networks controlling the second heart field and outflow tract development. So I'm just going to talk in a little bit more detail about one particular disease, congenital heart disease. This is 22q11 deletion syndrome, which I've spent a lot of time working on over the years, and it used to be known as DeGeorge syndrome. It's the commonest human interstitial deletion syndrome, with 1 in 4,000 live births, and it's due to haploinsufficiency of a part of chromosome 22 in a sort of 1 to 3 megabase region, which you can see down here. Now, it's a bit confusing because you can get these deletions of 22q11 region of 3, anywhere between 1 and 3 megabases, containing, a large number of genes, but you get a very variable phenotype, which affects a number of different body systems, including craniofacial development, gland development, the brain, and also skeletal and renal development. And in particular, you get a very characteristic set of cardiac defects, which mostly affect the outflow tract and great vessels. So common arterial trunk, double outlet, VSDs, interruptions, and so forth. So essentially affecting the second heart field. This phenotype is very variable in patients. So one patient might have a very severe cardiac defect and the next might have some mild childhood fitting relating to calcium regulation. And yet they may have, the first severe patient may have a much smaller deletion than the second patient. And so it's not possible to map the severity of the phenotype to the size of the deletion, which makes life kind of complicated. What people have done over the years is narrow down the region of that's commonly deleted between all these patients. And then people have gone on to use animal models to try and work out the function of all the genes involved and how they're interacting with each other. And what's became apparent quite quickly is that TBX1 is always in the deleted regions. This is a transcription factor that we know is very important for cardiac development. Over the years, a few non-deleted patients, so patients with 22q11 deletion syndrome, but without the 22q deletion, there have been some mutations found in TBX1. So TBX1 is the sort of major candidate gene for 22q11 deletion syndrome. And we've used mouse models to really understand what's going on here. And the first thing that we find is that the defects are dosage dependent. So if you knock out one copy of TBX1, then you end up with these really characteristic second heart field defects in about 50% of the mice. Sorry, not 50% of the mice, you've got 50% of TBX1 here because it's heterozygous deletion. And you get interruptions, you get abnormal right subclavian, so here it's a retroesophageal double outlet and so forth. If you knock out both copies, then you get common arterial trunk and VSD. And we've made a sort of range of different alleles where we knock out different amounts of TBX1. And we find that this particular one, TBX1 neo2 neo, which is a hypermorphic allele, which has about 20% of the normal amount of TBX1, is the best copy of human 22q11 deletion syndrome. And those mice will show, or embryos will show, the range of cardiac defects that you see in human patients. From these studies, we've also found out that TBX1 is required in the second heart field for progenitor cell proliferation and therefore outflow tract development. And although it's not expressed in cardiac neural crest behavior is abnormal in these mutant mice. So signals controlled by TBX1 are important for neural crest development. It's also expressed in the pharyngeal and endoderm and ectoderm, and those signals also play into normal crest and second heart field development. So this process is complex. It's doing a multiplicity of different things. It's controlling a whole set of downstream genes, including FGF8, FGF signaling, Notch pathway signaling, the retinoic acid pathway, and a whole set of other things. Then one of the other sort of major human syndromes that we see, you have mutations in JAGID1 and NOT2, which are the ligand and receptors for the Notch, or one of the ligands and receptors for the Notch pathway, and this underlies Alagille syndrome, where the cardiac phenotype ranges from a mild pulmonary stenosis to tetralogy of Fallot. This is just a schematic of the Notch pathway. So you get signaling, you get cleavage of the intracellular part of the Notch receptor, which moves into the nucleus, de-represses target genes, leading to activation. And in mice, if you inhibit this signaling in the second heart field specifically, then you give rise to defects that are very similar to what you see in Alagille syndrome. And it's also necessary in outflow tract cushions and cardiac neural crest. And if you delete notch signaling in cardiac neural crest, then you get a range of tetralogy of Fallot-like defects. And then finally, just to quickly mention the epicardium and epicardial EMT. This is regulated by a whole set of signals, particularly focusing around Wilms tumor 1, which is a major genetic regulator of epicardial EMT. So it regulates snail and the genes necessary to induce a mesenchymal phenotype. And retinoic acid is also very important in this process. These genes again being regulated by WT1, which in turn regulates a whole set of signals that go down to the myocardium. And there are also signals from the myocardium important in regulating the epicardium. Okay, so I'm sorry, I don't have a clock. How are we doing for time?
We do have actually 30 minutes left.
Okay, great.
Including time for questions.
Okay, super. So
maybe do the heart generation and then.
Yeah. So basically I've got a few slides on heart regeneration and the role of, and then I've just got a few slides on the sort of research project, just to give you a kind of idea of the sort of output that you get when you do these sort of developmental biology studies in the lab, which Emma knows because she's produced sort of work herself, but just to give you a little flavour of that kind of thing. So it probably won't take me very long to go through this, and then we'll have plenty of time for questions. Okay, so heart regeneration. You know, at first glance, you think, what on earth has developmental biology got to do with regenerating a heart? And this slide is really, you know, this is when I talk to students who are not medics and don't really know very much about cardiac disease. But essentially, you know, as you know, cardiovascular disease is one of the leading causes of mortality in countries around the world. So, you know, in the UK, 25% of deaths, 1 every 3 minutes. And there are millions of people living with the consequences of heart disease, and the cost is enormous. So, you know, one of the major causes is coronary heart disease, potentially leading to a heart attack due to occlusion of the coronary vessels, which will then lead to the death of the muscle supplied by those vessels. And even if you survive a heart attack, and a lot of people do, then the chances of developing heart failure afterwards are very high. And this is because adult mammal hearts do not regenerate, or at least they do very, very slowly. Their cardiac myocyte turnover is very low. And instead of regenerating healthy muscle, you get fibrosis and scarring. And this scar kind of forms a patch within the injured heart. And it doesn't contract normally because it's not formed of muscle. That means that the heart cannot beat properly, and therefore blood flow is not normal, and you go into cardiac failure. So because of all of this, there's a lot of research at the moment into therapies to promote replacing lost cardiac muscle and also to improve the blood supply to an affected region. And there's two kind of different strands to this. One of these is cell-based therapies, where the idea is to try and put back in either cardiac muscle or cells which will give rise to cardiac muscle. And this is a sort of cell transplantation approach. And it's aimed at replenishing lost cardiomyocytes. And there are a number of different ways of going about this, but obviously iPSCs and induced pluripotent stem cells derived from the patient, or ESCs, to produce cardiac progenitors, which can be differentiated and put back into the patient, are the focus of a lot of research. And there are beginning to be some progress there, which I'll talk about in a sec. But obviously there, particularly where the new cells are not derived from patient tissue, there are issues to do with immune rejection. There are also problems to do with being able to manufacture and isolate enough cells of the right type. And then obviously there's a large amount of clinical regulation around this and complying with all of this and making sure that these therapies are safe for patients are also, quite challenging. The other side is trying to develop cell-free therapies. And these are based on the idea of directly stimulating endogenous cardiomyocyte production through a variety of different methods, be it drugs, engineered viruses, DNA, vectors. And the idea is that you'll stimulate pre-existing cardiomyocytes into proliferating. Excuse me. And one of the sort of big ideas here is reactivation of developmental pathways that regulate cardiomyocyte development during normal development. If we can do that in a disease setting, then this may act in a therapeutic fashion. And a lot of the research is directed towards looking to see what is different between models where cardiac regeneration takes place. So in zebrafish or neonatal mice, they will repair their hearts. If you cut a lump off it, they will replace them. But this does not happen in adults. And if we can work out what those differences are, try and recreate them in the disease setting, then this may have a therapeutic effect. The other area where there's quite a lot of work, and this links back to this idea of reactivation of development pathways, is trying to improve neovascularization of the injured region, which improves circulation to the injured region and therefore will support regeneration of cardiomyocytes or any sort of therapeutic attempts. And also importantly, there are a lot of becoming more and more apparent that both from the cell transplantation and cell-free side of things, paracrine signaling is extremely important in improving cardiomyocyte replacement therapy. So we need these signals that may come from transplanted hearts or from the addition of chemical compounds or whatever. So this is, just a couple of examples of the sort of research that's being done here. So in this particular study, the authors used induced pluripotent stem cells to make cardiomyocytes, which they did using a monkey, and made sure that they had the same immune characteristics. And these cardiomyocytes, which were grown in a dish, were injected back into the monkeys after a myocardial infarction. And you can see here in green, the iPSC cells have really integrated well into the host, and they're supporting recovery from a myocardial infarction. And you can see that these cells are making structural cardiomyocyte proteins, so sarcomeres. You can see here. Oops. And there's also good integration of with endothelial vessels, endothelial cells, so blood supply. And they found that these grafted cardiomyocytes survived for 12 weeks without any evidence of rejection, and that there was electrical coupling with host cardiomyocytes, so good integration. And there was, most importantly, improved cardiac contractile function at 4 to 12 weeks after transplantation. However, there was an increased incidence of ventricular tachycardia compared to controls. And some later research suggests that that's perhaps because some of the transplanted cells don't quite have the same maturity of cardiomyocyte differentiation, and that's an area that's still under investigation. So this slide sort of compares those species which will regenerate their hearts with those that don't. And we can see that those that do re-express these genes that are important in early embryonic cardiac development following an injury, whereas these species don't. Instead of re-expressing these genes, you just get this fibrotic scar and abnormal remodeling. And it's important to know that if you remove the apex of the zebrafish heart, within a day, you start to see re-expression of these genes. And you get this sort of this blood-filled clot that stops, protects the injury, stops it bleeding. This slowly resolves into fibrin, and then bit by bit, the fibrin is replaced by regenerating cardiomyocytes until 2 months after the injury, the heart is completely repaired, which is quite impressive. And people are very much focusing on trying to understand what role these genes and others are playing in this recovery and how we can use that in a therapeutic fashion. Other work includes investigating secreted factors such as thymosin beta 4. So work that was done a while ago found that if you add thymosin beta 4 to adult epicardial explants that don't normally undergo EMT, then you can induce them to do so. Which means that potentially you can use thymosin beta 4 to stimulate the replacement of coronary vessels in an injury setting. And in fact, the authors went on to prove this in mice, where they treated mice with TB4 before inducing a myocardial infarction. And they found that following that, the mice that had been treated with TB4 activated epicardial cells, had a better recovery, and in some cases, potentially, actually, those activated epicardial cells gave rise to cardiomyocytes. However, this is quite controversial and some people disagree. So that's an area where work is still going on. Thinking about paracrine signaling, follistatin like 1 has been shown to be really important here. So this is a secreted factor, and in the epicardium, it's got potent cardiogenic activity, but you lose this expression after a myocardial infarction. I'm using the pig as a model because pigs have got a very similar cardiac system to humans in terms of size, metabolics, heartbeat, and so forth. What people have found is if you take a collagen patch and you soak it in follistatin like 1, produce a myocardial infarction, then put the patch over the infarcted region, then it will promote the regeneration of pre-existing cardiomyocytes in both the pig and also in mouse models. So this is obviously a potential therapeutic avenue. And then very recently, people have used, so Sawa and colleagues have used induced pluripotent stem cells to create sheets of human muscle cells. And they've used these sheets as patches in a similar way, and they've put them onto pig hearts that have undergone myocardial infarction. And they've shown that doing this can improve the organ function. However, cells don't seem to integrate into the heart tissue, particularly when applied like this. And instead, they think that this improvement is due to the cells releasing growth factors and other paracrine factors that will help endogenous regeneration of the damaged muscle. And very recently, there's been some news suggesting that this approach is actually being applied in the first clinical trials in Japan, 1 or 2 patients. Okay, so last few slides, just talking about the research that I do. So I, for a long time, worked on 22q11 deletion syndrome with Pete Scambler. And we're particularly interested in identifying genes downstream of TBX1 using our mouse models. One of the pathways that we've identified as being greatly dysregulated is the retinoic acid pathway, which you can see here. So you make retinoic acid, it's a small lipophilic molecule, and it has a couple of interesting, so it will signal in a sort of diffusible fashion to neighboring cells, or it will enter the nucleus, bind to receptors that are bound to target genes, remove co-repressors, replace them with co-activators, and lead to the activation of target genes. And because of its diffusion properties, you get a sort of gradient across the embryo within particular tissues, which regulate this transcriptional program. So it's very important for programming the embryo. And how much retinoic acid is available for this is regulated by a couple of different enzyme pathways. So we make retinoic acid from dietary precursors. We see the embryo, this is relying on maternal diet. And the precursors, this is a vitamin A, basically. So once we've got vitamin A, retinol, into the cell, it undergoes a couple of enzymatic processes. Firstly, giving rise to an intermediate product called retinaldehyde, and then the retinaldehyde dehydrogenases convert this into active retinoic acid. We've got a second set of enzyme family called CYP26s, so cytochrome P450, 26. There were 3 of them in embryogenesis, and they degrade retinoic acid before it gets into the nucleus and make it less biologically active. And between them, they regulate how much retinoic acid is available throughout an embryo. In our TBX1 mutants, we found that CYP26B1, as along with the other CYP26s, was altered, downregulated, not expressed in the normal way. So we wanted to know whether this was actually meaningful. And in conjunction with our collaborator in Japan, we got hold of the CYP26B1 knockout mouse, and